No, thanks a lot to the organizers, and let's take a look at the time, and then I'll try to stick to 30 minutes, if not a little bit faster. So I'm going to tell you all today a little bit about our work going on at DAISY, where I'm leading a small group on time-resolved crystallography of photoliths, which hopefully you will enjoy. Let's see, maybe it doesn't work. Yeah, it wants me to identify my keyboard and stuff like this, so no problem. I'll just hit the space bar. OK, so my talk really begins with the sun, which is a topic that we've heard about a lot already today. So as we all know, and we've heard about earlier, the sun, and specifically sunlight energy, fuels biology as we know it, and also our civilization, of course, through the fossil fuels that we use to power everyday life. And so the sunlight energy is a really great thing. But of course, as many of you may have experienced, I know I have, sunlight can also be quite dangerous, right? So if you receive too much radiation from the sun, especially in the UV part of the spectrum, it can end up with kind of nasty results. And one of the most common and dangerous forms of damage that we and all organisms can receive from sunlight is to our DNA. And so, of course, DNA is made of this very beautiful helix of conjugated systems, conjugated bases, which absorb very strongly in the UV and are susceptible to UV-driven chemistry. And here I'm going to show two different lesions that can form when sunlight impinges upon DNA, or when UV light impinges upon DNA. And so you can get these chemical cross-linking events where, especially if you have two pyrimidine, for example, two thymine bases that are sequenced adjacent in this double helix, you can basically get photochemical cross-linking that will link these two thymines or pyrimidines together into the so-called cyclobutane pyrimidine lesion, which has these non-native double bonds here, or two bonds here, sorry. Or you can get these so-called 6,4 photoproducts, for example, where you have a sort of more complicated linkage between these two bases. And there are actually more esoteric photoproducts, but these two account for almost all of the DNA cross-linking events that will be present under normal conditions, normal sunlight. And these CBD lesions account for about 80% of all these photochemical cross-linking events. And if you get one of these in an E. coli, if you're an E. coli and then your genome is replicated, each of these lesions has about a 7% chance of inducing a mutation downstream. So as you might suspect, linking together the bases in this kind of non-natural way distorts the structure of the DNA and therefore impairs the ability of the replication machinery to read it when the DNA is copied. The 6,4 photoproducts are less common. They're about 20% of all the lesions, but they're much more dangerous, so that's worth noting. And in case you were to think that these lesions are uncommon and you have nothing to worry about, I have some bad news for you. So for example, in humans, it's been estimated that on a very bright summer sunny day, somewhere at sea level, you might receive about 100 of these lesions per skin cell per second. So all life that's living under the sun or exposed to sunlight needs to repair this kind of DNA damage very rapidly in order to survive. That's true for us and all organisms. And so we're studying one way that this DNA can be repaired. It's just an enzyme called DNA photolyse. It's a photoenzyme, like Darren mentioned this morning. So it uses light to actually elegantly repair this light-induced DNA damage. And these enzymes are very ancient, so they're present in all kingdoms of life. And there's a family of them that have evolved over time to different situations. And the family actually also includes the cryptochromes, which are a flavin-based photosensory family, primarily photosensory family, though they maybe have other functions beyond that. And so this is just to say basically that we focused our studies, and today I'm going to tell you about a particular one of these photolyses. It's a CPD photolyse, so it repairs these four-membered rings that I showed on the previous slide, or like you can see here actually. There are other photolyses that are specific for those 6,4 lesions that I showed. So I'm not going to talk about one of those. and it's a so-called class 2 photolyze. And you don't need to worry about this. This is just an evolutionary classification. But the main point is that, in some of the numbers, the timescales and stuff that I'm going to talk about, there is some variation between these different systems. And so to orient you to our beautiful enzyme here, this is one of the crystal structures that we solved. This is just the dark resting state. And what you can see is that the photolyze is able to specifically recognize these DNA lesion events. And so here we've co-crystallized it with a strand of DNA that includes a synthetic lesion that we've been able to incorporate at a very specific site in order to produce high quality, well-diffracting crystals. And it basically flips this lesion out of the double helix and positions it proximal to an FAD cofactor that sits here that acts as the primary driver of the chemistry of the system, and also the chromophore that accepts the photon in order to initiate chemistry. And let's see, right. In all known photolyzes, there's also typically an antenna chromophore that's present, and this is a secondary chromophore that is thought to broaden the wavelengths that the photolyze can use to actually do its chemistry, because the flavin, this FAD cofactor, is in the fully reduced form in the active photolyze, and so only absorbs in the far end of the blue spectrum. And the secondary chromophore broadens that to encompass most of the visible spectrum. But it's worth noting that in our study, this chromophore is absent because actually the E. coli that we express the system in don't produce the chromophore that this system uses. But in some ways, we were happy with this because the system is chemically active with no problem as long as you excite it with the blue end of the spectrum. And actually, basically all the spectroscopy work that's been done on these systems have purposely removed these antenna chromophores so that they can probe at those wavelengths. And so we were able to directly compare to those previous studies. If you look at the active site in a little more detail, you can see that actually the flavin is in this kind of unusual conformation. It's unique to this family of enzymes, so unique to the photolyzes and the cryptochromes, in which the adenine part of this FAD molecule is actually inserted here between the so-called isoalloxazine, which is this three-membered ring that acts as the primary, basically the primary chromophore, and as you will see later, an electron donor, and the thymine dimer. So it's just worth keeping in mind that, yeah, there's this unusual and very interesting conformation of the chromophore, and maybe, I'm not sure, but Tatiana will talk more about this later, so it's also worth highlighting for that reason. Kind of other notable features of the enzyme are that it's got this nice positively charged surface that's perfectly complementary for the DNA, and you can see the active site pocket here for the DNA double helix to basically nestle in perfectly, ready for action. And so when we began our work, actually we knew a lot about this system. It had been very well studied for a long time. There's already a Nobel Prize given in part for this to Aziz Sankar. And a lot of the previous work came from spectroscopy, but it was also backed up by computation, it's worth saying. And so I'll just quickly tell you sort of what we knew when we began our work, and then, try to highlight some of the open questions that we aimed to answer with time-resolved crystallography, which is where we came in. And so here's the basic mechanism as it was understood when we began. You have this flavin species, so this is the sort of active part of FAD, as I mentioned earlier, and then of course, the thymine lesion here. So we studied a thymine dimer lesion exactly as shown here. And the reaction begins with photoexcitation. So if the flavin absorbs a blue photon, in our case we used 400 nanometer excitation, then this can create this excited state that's capable of basically undergoing an electron transfer reaction to the thymine. So the flavin will donate an electron to the thymine, and it's known from chemical studies that actually the reduction of this thymine species is sufficient for repair. So as long as you can get an electron onto this thymine dimer, then it will spontaneously split. So it will form an anion and then split. And actually these two bonds will be broken, and the aromatic systems of the original thymine bases will be restored. And then now you have still an extra electron on the thymine species, and that can be actually back electron transferred to the flavin that restores the flavin into its original state so that the cycle can continue and the enzyme can undergo another turnover. So we have the restored flavin in a fully reduced state, which is also called the hydroquinone state for sort of the flavin nerds, the fad nerds out there. And then now, the fully restored healthy DNA. So that's good. And the time scales for this were, of course, known from things like transabsorption spectroscopy. And so now I want to tell you a little bit about what open questions we targeted with our work that were uniquely answerable by time-resolved crystallography. And I mean, really, at the heart of this enzyme is a high quantum efficiency. And that's something that we're really interested in studying in as much depth as possible and is an ongoing topic of our work, actually. And so the photolysis, the exact quantum efficiencies are a little bit debated, and probably it varies from exactly what photolysis you're studying, like I showed on that previous slide. But there are numbers that are reported in sort of the 50 to 90% range. That just means, right, maybe about half or more of the photons that the photolysis actually absorbs can result in a productive DNA repair event if there's that substrate present. And a lot of what we know about these systems actually comes from these chemical models, like I'm showing here, especially pushed by Thomas Carell, who did a lot of really beautiful work in this field. And in these chemical systems, they're quite interesting, but basically what Carell and others did was take these thymine dimers, or sort of very close relatives of these thymine dimers, and just covalently link flavin or flavin analogs to them. And if you do this, you can actually get DNA repair activity upon illumination. So these flavin analogs are capable of transferring electrons to these thymine dimers and splitting them. But the quantum efficiencies of these chemical systems are universally low. So typically, they're much less than 1%. In perfect conditions, the highest I've ever seen is around 2% maximum. So if you tune the solvent and the chemical nature of this to something like this, which is obviously fairly carefully engineered, you can achieve sort of 2% efficiency. So it's clear that the enzyme has a big advantage in this regard in terms of using light to do DNA repair chemistry, and we want to understand how that's possible. And so we can break this down one step further, which is that if you think about what happens after you captured this excitation energy, so you've absorbed a photon and your DNA photolyzed, there are basically two ultimate fates for this energy, if you think about it coarsely. In the first fate, you can basically reach the ground state once again through either some radiative or non-radiative decay. So basically, you dissipate this excess energy as heat or light. The light-giving channel or the radiative channel in these systems is very small, so almost all of it would be in the form of heat. Or, of course, you can drive that electron transfer chemistry that you want to the flavin, or to the thymine dimer, excuse me, and thereby do DNA repair. So you have the competition basically between this de-excitation back to the ground state and the favorable chemistry forward. And we can get some insight into this by just studying this flavin species in isolation and looking at how quickly does it decay back down to the ground state. So if we look at just FAD in solution, what we can see is that decay is very, very fast. So this is some work from Dongping Zhang's lab quite some time ago that basically shows that the free flavin in solution decays in the order of picoseconds, or tens of picoseconds rather. And so this channel is very fast. Basically, the de-excitation happens very rapidly. And that doesn't leave very much time for this productive electron transfer. And in many ways, that's what results in the low quantum efficiency of these chemical systems, which are basically in solution, right? So they exhibit the same sort of phenomenon. If we look at the enzyme in contrast, that excited state lives for quite a bit longer. So typically in photolysis, this excited state lives on the order of nanoseconds. And that means that there's just a lot more time for this productive electron transfer to happen. And if we break it down in terms of time scales, what you can see is that, as I said before, that flavin in solution decays on the order of sort of tens of picoseconds. The flavin in the photolysis without any DNA around is order 2 nanoseconds. And the electron transfer time scales that are measured in photolysis when the DNA is present are on the order of hundreds of picoseconds. And so you can see that that lifetime of the excited state is really necessary in order to provide sufficient time for this forward electron transfer. And so what we're interested in is, okay, then if we know that this stabilization is important, how does the enzyme actually provide that stabilization of this transiently populated excited state? And so we're interested in probing the geometry of those excited states in order to understand this. And that's a question that can be answered by time-resolved crystallography at XFELS, as we've heard before. The second question that we had was a little bit easier to understand. And it basically involves the mechanism of the DNA repair itself. And so this was debated for quite a long time as to whether this lysis of these two bonds that need to break in order to restore the thymine bases to their native healthy form, whether that lysis occurred in a concerted mechanism, which basically means the two bonds break in a way where there's no barrier in between them, so there's no intermediate in between these two, or via such an intermediate where one of the bonds was formed and you had this metastable, transiently populated one bond anionic radical species, which would imply that there's potentially a small but present barrier along this pathway. And this had been debated for quite a while. I would say there was probably significant evidence, but not conclusive for this one bond intermediate. But all of these spectroscopy and computational studies were challenging to interpret or to adjudicate between, and this debate basically continued. And of course, that's another question that we can answer with time-resolved crystallography, hopefully by looking for this one bond intermediate and seeing if we could capture it. The final question that we were interested in is a little more biophysical in nature, so less chemical, and it involved product release. So if we look at the timescales of the chemical steps of the reaction, basically you have photoexcitation, electron transfer, and this final thymine splitting. They range from sort of the ultra-fast to the nanoseconds. So all the chemistry is done in nanoseconds, right? But the limiting step of the entire enzyme turnover cycle is actually product release. That's 10 to the 4 decades in time slower. And so this is what actually limits, in some ways, under bright light conditions, and when there's a lot of light and there's a lot of substrate around, how fast you can repair DNA. So you can think about quantum efficiency as kind of limiting under dim light conditions. That's how efficiently you can use a light. And under bright light conditions, turnover is what's limiting. And you might think, okay, that seems a little bit silly, like this is so much slower. But actually, it's remarkably faster than the rate at which the enzyme releases substrate, which of course is another way of saying, right, that the enzyme has a preference or binds substrate much more tightly than it binds product. And to me, this is quite interesting because when you think about what differentiates product and substrate, it's only two small bonds in these two thymine bases here. But of course, we can see that the enzyme binds the DNA using this large surface, right? And so how is it able to effectuate this big discrimination between substrate and product just by looking at this sort of local region of change? And that was another question that we thought time-resolved crystallography could address. So we had these three primary driving questions. I mean, I'm not going to say it's an exhaustive list, but they were the things that we were the most interested in going after if we knew the positions of the atoms as a function of time through a time-resolved crystallography experiment like we heard about this morning. And so we traveled, actually, this work that I'm going to talk about was done at the Swiss file. Here's an aerial photo of the Swiss file. It's about halfway between Basel and Zurich. Many of you probably know it. And it's built into the forest here. It's a very nice place to go do experiments. We were very lucky to work with the Alvra team, who did a great job of supporting our work, especially since basically the entire team from CFEL, from Hamburg, had COVID during the experiment and couldn't participate. So there were sort of three of us left standing by the end of the beam time. You can see everybody else is in masks. And yeah, this was in 2022, so still quite fresh in terms of COVID fears. But we managed to have a good experiment. We collected 10 time points spanning 3 picoseconds to 100 microseconds, highly redundant, and we had a small power titration as well. And so now I'm going to go through each question again and try to answer it in the form of a movie. The movies go through these different time points that we collected, and you'll see that actually they basically morph one into another. So between the different time points, there's some interpolation. Obviously, that's Hollywood. But I think that you'll be able to see the scientific content quite clearly, and this is a nice way to represent it. And we'll start by looking at the sort of fastest timescales and addressing what that geometry of the excited state looks like. And so here, we're zooming into the flavin, and you'll see a flash that sort of represents the laser excitation. And for me, it's interesting to also watch what the residues around, how they basically move in order to accommodate the structural changes that basically are driven by this photon absorption. And so what we can see is that there's this kind of butterfly bending of the flavin where it moves from a sort of upwards geometry. I have it on the next slide. In the ground state, like this. I mean, it depends which way you orient it, but to me it's upwards. And that's a well-known feature for fully reduced flavins, that they have this kind of characteristic bend in many, many flavoproteins, to kind of an inverted. So in some ways still a lot of sp3 character around the central axis, but now pointing down at 3 picosecond pump probe delay, which is we know that the flavin must be in the excited state at this point. And what we can see is that those residues that are surrounding the flavin site actually are able to move in order to accommodate this structural change. At later time scales, we can see that this ring system planarizes, and that happens after the electron transfer process should have taken place, for example, around 3 nanoseconds. So there's quite a few time points that we have that exhibit this planar geometry. And so now we've captured actually, I mean, to me this is amazing. Like what we're looking at potentially is the geometry, right, of an excited state. And we've been able to capture that using this time-resolved crystallography. And we're aiming to follow up to understand how, this has really ignited new quantum chemical calculative work in our hands that we're using to try to understand what is the electronic structure that this thing has, and how does that stabilize this excited state given the enzyme structure that's all around. Second question involved the lysis of these bonds. So I'll play another movie that now goes a little bit further in time. So we have this dimer splitting that we're interested in. And we'll reorient ourselves now to basically look at this thymine dimer more directly. So you can see the 4-membered ring quite clearly here in the CPD dimer. And we'll go basically to 1 nanosecond pump probe delay, morphing between the different time points that we got. So here we are at 1 nanosecond intermediate, and if we look at the omit map, so ignoring the model but just looking at what the data show us, we see that we can model it fairly well with this 1 bond sort of intermediate structure, which is highly strained, of course, and once again probably far from quantum mechanically perfect, but that explains our data quite nicely. I mean, it's quite unambiguous that there's this 1 bond intermediate present. At longer pump probe delays, we can see that these now restored thymines become much more planar. And so in this case, I mean, again, I think it's remarkable because really what we've seen is chemistry in action, right? Bond breaking driven by this enzyme. And then we can look at the final question that we were interested in, which was product release. So what happens at the longest time scales that we've probed? And what can it tell us about how the enzyme discriminates that substrate from a product? So here we rotated back around to look outward from the active site pocket. And we can see the restoration of the thymine systems. And they actually dwell in this pocket for quite some time. But what we can see is that at long time scales, first we have ejection of this thymine and then this thymine. And what's quite remarkable is that while we have a decent amount of geometrical rearrangements inside the active site pocket, there are relatively few changes to the protein structure outside that pocket. And so the discrimination is truly local. And we believe that we can describe or explain this as follows. In the substrate form, we have a non-aromatic system with these non-native bonds basically pulling these two bases together. But once these bonds have been split, we have two aromatic bases that want to base stack just like they would in the wild type DNA. But there's not enough room for them in the active site actually. So they basically push against one another and swell the active site pocket. This actually causes the rupture of a few hydrogen bonds. But in some ways, it looks like the geometry, actually, the strained geometry of these two bases that are pushed too close together is a primary driving force for their ejection. And the change is actually in the DNA, I should say, not the protein, that clearly drive this product release, which to me was a surprise. So we have these three questions, and I hope that you found the movies interesting. There's, of course, many unsolved mysteries still for photolysis. I mean, even for the CBD photolysis that's been studied for quite a long time. The first one that we're going after is like why this, again, going deeper, one level deeper, why this bent fad is so stable. But now we have that structure to be able to ask this question in a precise way. Again, I mean, I'm quite interested in, people have been studying this for quite some time. Why is this adenine inserted between the donor acceptor? Again, maybe Tatiana will talk about that tomorrow, I'm not sure. So stay tuned for hers. And then another really interesting thing is the role of water. So we see water rearrangements on ultra-fast timescales that accompany this bending motion of the flavin upon excitation, and also in the active site pocket at all stages of the repair reaction that are difficult to understand and actually difficult to see in some cases in our structures. And with that, I'm happy to take questions, but I'd just like to quickly say thanks to the huge team actually that came together. But really in a lot of ways this was a partnership between me, but driven by Nina, who is a postdoc in our group, and is the one who was really responsible for driving this work to completion. So big thanks to Nina. And thanks for your attention. I'm happy to answer questions.